Hi, everyone. We appreciate you taking the time to join us today, and I hope that this presentation inspires you as we kick off a new year. We are really excited about the promise of great things to come in 2023 and to be presenting to you today about molecular biological tool game changers and how MBTs have really impacted our decision making for the past 30 years. Then we're going to talk about what's happening to peer inside that black box today, and finally, what we're going to do to take it further apart in the future. You know, over the past 20 years, I have done a lot of presentations, and I have to say this is one of my top 5 favorites, so I hope that you will be excited by this too. Before we get started, just a couple of housekeeping notes. Please note that this webinar is being recorded and it will be available in the resource section of the Microbial Insights website within the next week. A link will also be emailed to everyone who registered for the webinar, so you don't have to contact us. That's going to be automatic. And after the presentation today, we're going to have a short question and answer session. So feel free to type your questions throughout the presentation into your GoToWebinar software panel, and we're going to answer as many of those as possible at the end. So the lineup for our presentation today consists of 3 speakers. I'm Dora Taggart, CEO of Microbial Insights, and I'm going to start talking about the milestones that we've accomplished over the past 30 years. Particularly, I'm going to focus on the 22 years that I've been here with the company. Then Dr. Kate Clark, the Vice President of Global Laboratory Services for Microbial Insights, will show you where the tools are today and how critical that they are for today's site assessment. And then finally, Dr. Sam Rosalina, our VP of Applied Innovation here at MI, is going to take us home and end the talk with the research and development that he and his division are doing to really propel us into the future. You know, the past 31 years of Microbial Insights have been nothing short of exciting and revolutionary. When Dr. David White founded our company, it was his ultimate desire as both a medical doctor and a PhD to take tools that were developed in the medical industry where they have funding and help us to apply them to site cleanup and remediation where our funding is so limited. And when we would talk to him at dinners, he would say he really wanted to find a way that we could actually talk to these microorganisms so we can find out who they are, what power they have, and then be able to harness their natural power to make site decisions more cost effective. So when I was thinking about our talk today and the course that we've been on over the past 31 years, I thought about the 1 degree rule, which is critical for aviation. This rule states that for every degree a plane veers off its course, it'll miss its destination by 1 mile for every 60 miles that it flies. And this rule reminds pilots that even a change of 1 degree can make a huge difference. So if we're off course, understanding the direction and correcting even 1 degree at a time can help us get to our intended destination. Small changes really have big impact. And I think the same applies for us in site management. If we understand the black box of microbiology and we start to talk to these microorganisms like Dr. White's always wanted us to, we can course correct so much more quickly. We can utilize the native power of the system and the products that our industry has created to enhance this natural power. And this knowledge that we attain can help us to start to answer so many of the questions that are present in every site assessment. And we need to remember that biology is a component of every corrective action plan. And we have come such a long way. You know, in the 1990s, we saw articles like these that were being published. The use of microbes is an emerging technology for remediation. That was in 1992. Then bioremediation of chlorinated solvents in 1993 is becoming a reality. And then finally, we see that in as late as 1998, there was an article that came out talking about bioremediation moving toward a credible technology. And this is the time period when Dr. White was founding Microbial Insights. MI was founded in 1992. And even back in those days, he was thinking about bioremediation and how to really understand the bugs that are naturally present. You know, before we had these tools, we had to go to a site and we would measure the chemistry and the geochemistry, and we would spend a lot of time trying to guess what the microbes in the black box would do if we added something to that subsurface. At that point, we still had so many questions, but with the evolution of these tools, we can start to see what's happening in the black box and start to really answer those questions. And I really love this slide that we're coming to. These tools have come such a long way. Originally, phospholipid fatty acid was the concept that Dr. White was using to help us get an aerial view of the community structure and what was happening. He was doing this long before the founding of Microbial Insights back in the early 1980s. Then we have the genetic revolution that brought about the ability to test for organisms that we were interested in. And as sequence evolution exploded, we became able to accurately quantify more and more organisms and genes of interest. Then we started utilizing in-depth chemical tools to help us understand the fractionation pattern of key compounds through things like compound-specific isotope analysis and stable isotope probing. And then if you look at 2022, now we have an exciting tool that can help us understand microbial interactions, and I think will eventually predict biodegradation using metabolomics. It's called the meta-array, and I can't wait for you to hear more about this today. So when we think about our site navigation and staying on course, if we're good navigators, we're going to take measurements that are associated with our chemistry and our geochemistry. We're going to add this tool for the microbiology using these molecular biological tools, and we're progressing leaps and bounds over where we were just 30 years ago. Now we've learned that chlorinated compounds aren't recalcitrant, they actually can be degraded. We also learned that petroleum hydrocarbons do degrade anaerobically, not just aerobically, and it's not ubiquitous. We can also add non-native bugs to a site and increase our rates of degradation. And finally, microbes can actually survive in some pretty extreme environments. We do see them in oil and gas pipelines. And there's so many more myths that have been busted with these tools. So what I want to do today is to go through a few of the tools that I think have really revolutionized our industry. As I mentioned before, Dr. White founded Microbial Insights on the use of phospholipid fatty acid analysis. This tool uses the cell membranes or the skin of the microorganism to give us broad characterization of the microbial groups. The benefit of this was that we did not need prior knowledge. We're looking at all the organisms present in the system, and especially back in the 1990s, this was critically important because we simply didn't know the pathways. More than 30 years later, we still see value in this tool, as it's a way to provide a fingerprint of the entire population and look for shifts in the community anytime we make a change. The genetic revolution is the next point, and that's really near and dear to my heart. This was really a huge game changer for looking at microorganisms. When I started with Microbial Insights back in 2001, we used phospholipid fatty acids, we used plate counts, we used a tool called denaturing gradient gel electrophoresis, or DGGE, and some of you may giggle when you remember that word. We also had some limited PCR that we used to investigate samples. But when the exciting work of well-known researchers like Perry McCarty and Frank Loeffler demonstrated that chlorinated compounds can in fact be degraded, I remember working with researchers like Sam Fogel as we tried to grow Dihalococoides on plates. It was a noble attempt, but an insurmountable challenge. Most organisms like Dihalococoides simply don't like to grow on plates in the lab, and we just can't replicate their in situ conditions. In fact, researchers have proven over and over, and we see this in publication, that less than 1% of organisms can be accurately quantified with plate counts. So we moved to something called qualitative PCR, and I remember sending reports to you guys which said plus, 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 plus for Dihalococoides detection, and that was based on the intensity of amplification that I could see in a gel. It was definitely better than plate counts, but still not accurate. Only a year later, in 2002, we were the first commercial lab to start offering quantitative detection of Dihalococoides, and this is still a critically important assay today. With this newfound gift of qPCR, we can start to design assays for numerous organisms and pathways that were important. So it quickly grew from our first assay to look at Dihalococoides to 20 assays to look at other genes and organisms and pathways, then to 50, and so on. Now today we have over 150 qPCR assays that are currently offered that have been developed. And as sequencing technologies advanced, so did our depth of knowledge of sequences. If you see these charts here, this is from GenBank. GenBank is a global housing database for sequences that researchers around the world use to deposit their findings. And what you can see in these charts is that from 1982 to present, the number of bases that were identified in GenBank has doubled approximately every 18 months. So this is really important for us to remember when we design assays for qPCR. You know, back in 2005, we developed an assay, this is just an example for you, of an important gene in anaerobic text degradation called benzyl succinate synthase, or BSSA. This was identified by Harry Beller's group. And in those days, our assay was based on those 4 sequences that you see on the left. By 2016, you can see how many more sequences had been identified just for this one gene. And those first 4 sequences were only a small subgroup. So today, rather than running one qPCR assay to quantify known BSSA genes, we actually sum together 8 different assays that we run to target that entire group. That's why it's critically important for labs doing qPCR to review their assays every year so we can see what researchers have discovered. And if there are new subgroups on that tree that have been identified, then we can redesign assays to capture more of those groups and expand our knowledge base. So developing so many assays and learning how many different genes and organisms involved in our processes is both a blessing and a curse. It was a blessing in that we have more depth of knowledge, but it required a lot of qPCR to understand a process. So in order to capture all of the knowledge with individual qPCR assays, it could be cost prohibitive. It was expensive and time consuming to run all of those singly. So back in 2012, I heard about a new platform for qPCR that would allow us to perform many different qPCR assays on a single array slide using through holes. Now this doesn't have the need for multiplex qPCR, which comes with its own series of concerns. Well, when we tested this platform, we found that we got the exact same results when we did it on these slides in those through holes as we do with individual qPCR measurements. So this milestone brought about a whole new suite of capabilities with this new analytical tool. And many of you have heard about the QuantArray platform. So there are now 5 different versions of QuantArray that you guys can order. For chlorinated solvents, we can quantify 29 different organisms and genes in a single analysis. So we capture not only Dihalococoides and other organisms involved in reductive dechlorination, but we also start to understand co-metabolic activity. For petroleum hydrocarbons, we know that there are a lot of genes and pathways for both aerobic and anaerobic degradation. And now with this, we can quantify 22 different pathways. And finally, for corrosion, understanding community interaction is crucially important for us to better mitigate risk that's associated with the microbiology in a system. So with this assay, we quantify 24 key groups of organisms that are important. And Kate's going to go into the depth of the last 2 that are really exciting. There's a QuantArray now that was launched in 2022 for natural source zone depletion and one for biogeochemical assessment. Another tool that I'm excited to talk to you about that greatly advanced our knowledge is called compound-specific isotope analysis. And this is one that Sam worked on launching back in 2016. And CSIA had been used by other industries for a very long time, but the use and remediation was limited due to inconsistent commercial availability. So back then, Sam helped us to bring this technology in-house so we could offer it at MI and have better control for the customers and make it more commercially viable. So by tracking the natural fractionation patterns of compounds, we're able to look at both biotic and abiotic mechanisms. And most importantly, we can get conclusive evidence of degradation for chlorinated compounds. It also allowed us to start to delineate our sites a lot better, and when we had multiple sources, to be able to identify those sources. In 2014, we launched our database of qPCR information, as Microbial Insights has the world's largest and most diverse repository of environmental samples. This database houses more than 25 years of qPCR data from over 150 different organisms and gene targets, from over 100,000 field samples from sites all over the world. We have samples from all 50 states in the United States, more than 45 countries on all 7 continents. So this database allows us to really put some context to that concentration data when you get those results. For instance, it can help us bust some myths. So if we look at a particular gene at a contaminated site around the world, we can start to understand the frequency of detection. So if we look at, for instance, around 10,000 groundwater samples that were analyzed for anaerobic text degradation using that BSSA gene that I described earlier, we see that only about 53% of the samples had detection. And out of over 30,000 samples for aerobic BTEX degradation, when we look at aromatic toluene genes that are involved in this degradation, they were only detected around 45% of the time. So this information really helps us to dispel the misconception that for petroleum hydrocarbon remediation, it's ubiquitous, the organisms are always going to be detected, there's no reason to even think about it. Well, we do need to, because there are problems that occur and detection is not always consistent across every site. We don't know if these organisms are present in every situation. And then finally, we can understand what a concentration actually means and put some context to it. Is 10 to the 4 high for this particular gene and organism compared to other sites and samples that are contaminated from locations around the world? And I don't want to fail to mention that we also have a database that was launched for the CSIA analysis, so you can start to research literature values, plot data, get easier visualization and interpretation. And I would really encourage you, if you're a customer of Microbial Insights, to use this free tool that you get with every sample that you do with us, and at least look at the data to see what it means and try to add some context for your reports. And our talk today would not be complete without looking at the amazing history of biotrap samplers. You know, Dr. White saw this tool at a conference in 1998, and he knew it could be impactful for the remediation industry. The biotrap was developed by Dr. Carrie Sublette at the University of Tulsa and DuPont. It utilizes powdered activated carbon and omics to form beads that are about the size of BBs, which have enormous surface area for organisms to colonize and grow. So the biotrap started to be used in the early 2000s as a passive collection device for microbes. They form biofilms within these beads, and you can see the cross sections there in the picture on your left. Then this tool grew, and we learned that we could amend it with 13 carbon compounds as a tracer of biodegradation during stable isotope probing. And this can be a way that we can prove in situ degradation of a particular compound when we trace that 13 carbon into the biomass and into the dissolved inorganic carbon. Finally, this was expanded into in situ microcosms to help us evaluate different treatment options in situ, so inside our monitoring wells, rather than having to pull water and soil and do bench scale studies. This tool has literally been used at thousands of sites around the world, and Kate's even going to show you a few of the newer adaptations that are being used today. And I can't end this section of the past without mentioning the word omics. Metagenomics that started with gel-based technologies like DGGE have changed. You know, when I did denaturing gradient gel electrophoresis years ago, we could identify 3 to 5 dominant organisms that were present in the gel. Today, we can use instrumentation to get more in-depth sequencing, identifying hundreds or thousands of sequences. which further our knowledge of specific organisms and genes, as we discussed earlier, and can help us get a deeper understanding of community interactions through next generation sequencing, shotgun sequencing, and whole genome sequencing. And as I mentioned in the timeline, at the end of 2022, we launched a new tool that I am fascinated by called the meta-array. This uses metabolomics to study the small chemicals called metabolites, which are found in biological samples. And I know for years I've been talking to you guys about the holy grail of proteomics and being able to analyze these complex proteins as they're formed so we can better understand what's going on at our site. But with all the research facilities and everything that's been done and the instrumentation that's been developed, we just can't get it reproducible enough for commercial processing and for it to be cost effective enough for us to be able to utilize this in routine site assessment. So I think metabolomics is going to get us there. That's going to be the metabolites that are formed during these processes. So if we can start to understand these, I think this is going to be the next step in the evolution. And as I was preparing this talk, I was thinking about how blessed I have been to see the evolution as I joined Microbial Insights back in 2001. I got to be part of the launch of the qPCR assays, which I feel were one of the biggest game changers in our molecular biological toolbox. Now, 22 years later, I'm beyond blessed to be part of launching a tool that I think will again revolutionize how we assess sites through metabolomics using our meta-array. And throughout the presentation today, you're going to hear a lot more about this tool, and it'll be described a little better for you than I'm doing here in summary, as Kate's going to show you what we're doing currently today with the meta-array data. And then Sam is going to share where we're going to go with it in the near future.
Okay, thank you, Dora. The tools that Dora discussed are still useful today, and our increased knowledge and technological advances have allowed us to continue to expand our capabilities to better address today's issues. With each advancement, we always keep in mind that we want to produce actionable data that practitioners can use to reduce uncertainty and course correct quicker. For example, qPCR is still the gold standard for quantifying taxonomic and functional genes. Along the way, we've added new qPCR assays as novel genes are identified or as a particular microbial process becomes more important to an industry we serve. High throughput methods like quantarray can reveal important pathways that weren't previously prioritized. Recently, we've introduced new quantarray suites to assess natural source zone depletion and biogeochemical processes. The complexity of the microbial consortia involved in both of these couldn't be adequately captured if we were just analyzing one or two gene targets. We've also continued to leverage our experience with biotrap samplers to develop new sampling techniques and even treatment applications. And then lastly, we're always looking for new methods that will give our clients a better understanding of the processes occurring at a site or in a system. And in a moment, I'll discuss our new meta-array metabolomics analysis, which can illuminate a facet of reductive dechlorination that we couldn't previously monitor. So I want to share with you a little bit about our new quantarray suites. The first one, quantarray NSCD, addresses natural source zone depletion of LNAPL. This is where petroleum hydrocarbons comprising the LNAPL are lost from the subsurface due to naturally occurring processes, including dissolution, volatilization, and biodegradation. This complex phenomenon is rarely accounted for in conceptual site models. And as Dora mentioned, anaerobic biodegradation of petroleum hydrocarbons was once thought to be insignificant. However, in natural source zone depletion, biodegradation is an important but complex component. It involves diverse syntrophic microbial communities, but by quantifying key microorganisms involved in natural source zone depletion, a clearer picture of site dynamics is revealed. Recent research suggests that natural source zone depletion rates in the field are faster than previously thought. This table is from a great review article by Garg et al. From Tom Sales Group. And as you can see, these rates can be substantial. So you don't want to overlook this possibility when you're developing your site conceptual models. This chart shows the interactions between chemical and geochemical parameters, which are shown in blue, and then the microbial groups and genes involved in natural source zone depletion pathways. And these are all included on the quantary, and those microbial groups are shown in the yellow boxes. So I won't go into too much detail today, but if you'd like a copy of this and the accompanying references, please email me afterward. I just want to highlight a few of the important microbial groups, starting with fermenters. Some fermenters in anaerobic L-NAPL zones are capable of biodegrading hydrocarbons to form hydrogen or acetate, which could be utilized by methanogens to form methane. So this relationship can help ensure that the hydrocarbon degradation process remains thermodynamically favorable. Then methanotrophs in the vadose zone can oxidize the methane produced in the saturated zone. Methanotrophs are also capable of degrading small chain alkanes and alkenes. We also have 4 qPCR assays related to biosurfactant production. These microbially produced surfactants can allow more NAPL to be released out of the pores and dissolved in the groundwater, making it more bioavailable. Once those hydrocarbons are dissolved or broken down into intermediates, quaternary petroanalysis is a complementary tool that can be used to assess their continued biodegradation. And this is an example of the results table for quaternary NSCD. So you'll receive concentrations of over 20 different gene targets. You can quickly see that the results in bold were detected. And the rest of the report includes heat maps and an interpretation guide to help you understand and apply those results. The second new quaternary suite assesses biogeochemical interactions, which can play an important role when we're working on complex sites and considering remediation strategies. So think about the geochemical parameters you regularly analyze for at your sites. How have you relied on that data to make decisions? What geochemical data do you consider to be reliable? Traditional analyses alone can sometimes mislead us. Many parameters are measured in the field, and dissolved oxygen in particular is a common culprit that may not make sense with the other data. Bulk measurements provide an average, but they don't necessarily reflect the microenvironment a microorganism is exposed to. So the plume model with our perfectly discrete redox conditions is just an ideal. There could be multiple respiration processes occurring simultaneously if the necessary electron acceptors are present. The quaternary BGC can help provide a clearer picture of the processes happening in the subsurface, and this is knowledge that leads to better strategizing. Understanding where your site is at with respect to electron accepting processes can be very helpful to assess the state of the site and develop a plan to shift redox conditions if needed. The quaternary BGC includes qPCR assays related to processes at every step in the redox ladder. Aerobic processes we can assess include oxidation of metals, sulfur, ammonia, and nitrate. If you're interested in the nitrogen cycle, the quaternary BGC is our most cost-effective method because it includes 10 relevant qPCR assays. And then if you're interested in a biogeochemical approach at a chlorinated solvent site, then a combination of quaternary chlor and quaternary BGC would be beneficial. So with that, it would include all the reductive dechlorination and aerobic co-metabolic pathways. And then the BGC suite would provide information on the potential formation of reactive iron sulfides. So this slide gives you an idea of what the quaternary BGC deliverable looks like. You'd receive quantitative results in concentrations that are reported in units of cells per gram or per mil. We consider order of magnitude differences to be significant, so we can quickly see that in this sample, iron-reducing bacteria are detected on the order of 10 to the 6 cells per gram. And that's several orders of magnitude higher than the other microbial groups we're testing for. Although there is a lot of functional diversity present in the sample, and we're detecting genes that are both involved in both aerobic and anaerobic pathways. We know that not everyone is used to interpreting microbial data, so all quaternary reports also display the results on a heat map to give you a general idea of whether the concentration is low, moderate, or high. The higher the concentration, the larger the population that could perform the process of interest and the more likely to see an effect. This heat map shows the same data from the previous table, and we can see the microbial group with the highest concentration is plotted in the green zone, and that's our iron reducers. There's also an interpretation section at the end of every report that provides descriptions of the gene targets tested. In addition to expanding our qPCR portfolio to meet today's needs, we've also continued to draw on our 20+ years of experience with biotrap samplers. The in situ bioreactor shown on the left can be deployed in existing groundwater monitoring wells to enhance biodegradation of contaminants in the aquifer. So this technology has its origins in the biotrap and uses bioset beads. We've been using bioset beads, which contain activated carbon, for decades as a tool to document contaminant degradation on biofilms formed on the carbon. The high surface area and porosity of the bioset results in a dense biofilm in the beads. This concentrates our indigenous degraders to as high as 10 to the 8 cells per bead. So we found that the same properties that make bioset beads an excellent forensics matrix also make it ideal for treatment application in this bioreactor. We've also introduced the MinTrap passive sampler that was developed in conjunction with Arcadis. Instead of microorganisms, here we're focused on collecting mineral precipitates in situ. And this is deployed in a monitoring well, so there's no need for drilling for soil samples. So this is a pretty satisfying before and after picture. You can see in the picture on the right that the black iron sulfides are collected on the silica sand sample sheet after the deployment period. So this sample sheet can then be divided and sent out for a variety of analyses. Okay, and I've saved the best for last in my section. As Dora said, we're very excited to announce the launch of meta-array metabolomics analysis. This is a low-cost screening tool. It's less expensive than a single qPCR assay, and it can be used to predict the health and activity of the entire microbiome. Metabolomics is the study of all small molecules within a sample, and it's proven to be a powerful tool when combined with the right statistical analysis. Meta-array is based on a growing database of metabolites and molecular features that allows for a broad screening of reductive dechlorination potential. The table and diagram shown here are taken directly from a meta-array report. Based on our model of field data from chlorinated sites, we can see if a sample's metabolic profile is indicative of a microbial community with high, intermediate, or low potential for reductive dechlorination. At the sampling event shown here, a little more than half of the samples showed a high potential for reductive dechlorination, while the rest fell into the limited potential category. We also have Dihelicocoides data for this sampling event, and one curious feature of the dataset is that some samples with Dihelicocoides concentrations on the order of 10 to the 3 cells per mil fell into the high potential group, and some fell into the low potential group. Here's a principal component analysis of the same sampling event, and you can see that our low potential samples separated into 2 different groupings. These 2 samples in the lower right come from locations with 10 to the 3 cells per mil of Dihelicocoides, while Dihelicocoides concentrations were much lower in the other low potential group. Here are the meta-array results plotted on the site map. The green check marks show wells that were predicted to have high reductive dechlorination potential, and the red X's show wells predicted to have limited potential. The main cluster of high potential wells are along the segment of the site that's undergone the most work. There was excavation, followed by emulsified vegetable oil soil mixing, and since then there's been more electron donor injected. There's also a ZVI addition through hydraulic fracking to get into the bedrock. So groundwater flow here is to the northeast, and the background well, which is MW205, showed limited reductive dechlorination potential, as we would expect. Down gradient monitoring wells, like MW208, are showing limited potential too. So the 2 wells circled in red are the low potential grouping from the previous slide with 10 to the 3 cells per mil of Dihelicocoides. Within the larger context of the site, this grouping makes sense because they're both bedrock wells and expected to share a fracture line. But if we're going by the Dihelicocoides data alone, the potential for reductive dechlorination would be indistinguishable between these 2 wells and the wells in the treatment zone that had a similar Dihelicocoides concentration. So we're very excited to have launched this analysis, and we hope that you'll find it to be a valuable screening tool for chlorinated solvent sites. Molecular biological tools routinely deliver data that can be reliably applied to enhance beneficial processes and curtail undesirable outcomes. We're at an exciting point where we no longer need to guess what's happening in the black box. We have decades of experience with molecular tools coupled with rigorous research on their application, so we can directly measure what's going on with the microbial community. This knowledge has been used in a variety of ways to keep projects on course. It can improve the accuracy of models, provide early alerts to potential problems, help make more informed management decisions, and identify transition points when a new treatment strategy is warranted or the switch to monitor natural attenuation is viable, all of which ultimately will result in cost savings for a project. So now I'm going to turn it over to Sam for a look into the future.
Thanks, Kate, and thanks, Dora. I love this webinar for many reasons, but I think it's especially great from the analogy that at Microbial Insights, we regularly tell people that trending the data is really important. And that's essentially what we're doing here today. We're looking at the past and we're seeing how far we've come. And so the next step is looking into the future. So we want to think about what are the black boxes in the future? What are the black boxes of our subsurface that we need to pull apart? One of those is agriculture. And I think probably many of you have seen this, but more than ever, scientists are seeing that the soil microbiome is playing a massive role in terms of how well plants grow, how much they produce, and how many nutrients are required. And not just scientists, but we're also seeing that a number of large-scale corporations like Walmart and even McDonald's are transitioning to sustainable agriculture because they're seeing cost savings, not just because it looks good, but they're actively seeing that they can use fewer nutrients, that their chemical demand for herbicides and pesticides are lower. And not only that, but they're bringing carbon back into the soil. And so there's less erosion and work needed to be done there. Not only that, but corporations like Levi Strauss and even Ralph Lauren are doing the same for cotton. And so what are the ways that we can course correct? If we're thinking about our 1 degree rule that Dora and Kate have mentioned, what are some of the ways that we can make sure that we're on track in agriculture? To us, the future looks, again, very similar to some of the other tools that we're looking at right now, and that's metabolomics. If we're looking at metabolic patterns within the soil, within the microbiome, we know that the biochemistry of these organisms are going to change, and the simple and both simple and complex biochemistry within them is going to change based on the environment that they're in. So if we can study those patterns, we can start to look forward and say, what are some of the predictions that we're seeing? We can start to look at the overall health of the soil. We can determine if the soil's been disturbed. We can determine whether nutrient deficiencies are occurring or whether we can save on nutrient runoff by applying it in a more conservative way. We can start to see if there's plant stress or disease prediction, and potentially even monitor carbon sequestration potential in the future. Something else that we're looking at is the importance of using metabolomics for reservoir souring. So many of you know that when a reservoir is tapped, when a well is put in for oil and gas, that the pressure from the reservoir itself pushes that oil to the surface initially. But once that pressure has run out, something called secondary oil recovery occurs. And that secondary oil recovery often uses something like water, especially if we think about the platforms that are out at sea. Water is used to push and inject it into the reservoir, which then pushes that oil up to the surface. And this can often result in an additional 40 to 60% of oil recovery compared to the original oil recovery. So this is a really important process. So when we're doing this secondary oil recovery by injecting water, often that water contains sulfate. In and of itself, sulfate is not a problem, but because there are usually sulfate-reducing microbes in archaea present, we start to see the production of hydrogen sulfide. Now, hydrogen sulfide can cost millions in revenue per reservoir, and often it takes a lot of money to negate. So in order to mitigate that hydrogen sulfide, additional processes need to be put in place to remove it. It can impact human health, and it can, of course, increase corrosion potential. Not only that, but there's product degradation through the process of sulfide formation. So it's really important that we tackle this. Now, the question often is, well, why don't we just analyze for sulfide topside? If we start to see hydrogen sulfide showing up at the top of our processing well, then we have an idea that hydrogen sulfide formation is occurring and that our reservoir is indeed souring. But the problem is, in many of these reservoirs, we have naturally present metals, and these act as sulfide scavengers. So these sulfide scavengers pull that hydrogen sulfide from the liquid, from the produced liquids, and instead keep it in the reservoir, forming things like iron and copper sulfides. And so those sulfides never make it to the top. Now, what this means is that there's no real way to monitor or measure reservoir souring until it's too late. By the time the hydrogen sulfide has used up all of the sulfide scavengers and has made its way to the surface, what we see there is that reservoir souring could have occurred for months and even years in some situations. So we're talking about saving lots of money by monitoring this early. So instead, what we can do is look at all of the organisms present. So if the sulfide reducing bacteria and archaea happen to stay within the reservoir and are not making their way up to the produced fluids at the top, then instead we can look at the whole microbiome in the same way that we're looking at the whole microbiome for our meta-array for chlorinated solvent sites. Because this whole microbiome is going to be affected by the environment of the reservoir itself, then these metabolic patterns are going to show up within them that we can monitor and measure and track and start to predict the onset of sulfide production and reservoir souring. So what we're doing is we're looking for sentinel metabolites. We're looking for these precursor biomarkers that can tell us, yes, this reservoir is starting to look like a soured environment, or even this reservoir has already begun to sour. From there, many mitigation potentials are possible, and it's just the key of finding that early enough to save money. So these metabolic patterns are really important moving forward, whether it's agriculture or souring, or as we're seeing groundwater remediation as well. On a similar topic, as Dora mentioned previously, we have a quant-array MIC, and that is focused on microbiologically influenced corrosion. So we know that there's kind of an estimate of current annual costs of corrosion to the oil and gas industry exceeding 60 billion globally. And tens of billions of those dollars are within the US alone. And an estimated 20% of all of these are attributed to microbial activity. So what we've found is that microbes are playing a much bigger role in corrosion and asset degradation than what we originally thought. And so this year, you're going to be hearing a lot about something called our joint industry project that we've joined with DNV, an ExxonMobil upstream research company. Already we have 5 participating companies across the oil and gas sector, and I want to throw out that we're happy to keep looking for more, but pretty soon we're going to be started. So we'd like for anyone to get on board as soon as possible. What we're doing within this is we're starting to look at biomarkers that are key to corrosion. Often these biomarkers, what we're monitoring, the problems that are existing, are happening within biofilms inside our pipelines or inside our processes, and they're difficult to reach and monitor. And so instead, what we're looking at are ways to find biomarkers within the produced liquid, within the liquid of the pipeline itself, that we can more easily monitor and track. And from there, we're going to be producing, not only identifying those biomarkers, key biomarkers, but producing key performance indicators that the participating companies within the JIP will have exclusive access to. So this is really important because we're looking at a much simpler way to monitor MIC and a much more predictive way to monitor MIC. So why do we think that this is possible? Well, we think it's possible because we've already done this. So this is a plot that is derived from a publication back in October of 2022 from Sven Lame and his group at ExxonMobil Upstream Research. This is important because we worked with ExxonMobil to develop an assay for a gene called MIC-H. And what has been found, both in laboratory testing as well as in field testing, is that the MIC-H gene can start to provide a much clearer picture of severity of microbiologically influenced corrosion within the pipelines themselves. So this is important because if we compare that to previous data of looking only at total cells through qPCR, we can see that our MIC-H biomarker is much more predictive. It can give us a lot more information about what's actively occurring rather than just looking at ATP or total cells. And I would be remiss if I also didn't mention, while we're talking about microbiologically influenced corrosion, that we have joined with Microbial Corrosion Consulting LLC and Scovis Bioconsult to form a full service alliance for microbiologically influenced corrosion. You can learn more about that on our website. And then finally, the big question, right? The one we're asked on a regular basis. What does the future hold for PFAS? We're seeing a lot of really important work happening. Many of you know of the work by Jaffe and looking at the A6 strain for degradation of PFOA and PFOS. We're also seeing some really cool work from the Loeffler group, wherein we're seeing transformation of fluorinated alkanes, but we're also being able to track that in the lipid bilayer. I particularly love this because it's reviving some of the lipid work that Microbial Insights was founded on. So it's very cool how science can sometimes come full circle. And then more recently, this has got a lot of buzz. The Trang and that group has shown that by essentially boiling perfluorinated compounds in DMSO at low temperatures, that they can start to degrade pretty quickly. Now, many people have said that this doesn't appear to have a lot of use in the field directly, and they may be right, but I think one of the key takeaways from this paper are some of the computational work that's been done. So through computational chemistry, we're seeing that some of the steps in the transformation of these PFCAs, these perfluorocarboxylic acids, have much lower activation energies than we previously thought. And so overall, what we're doing is we're looking at our 1 degree rule. And so far, we're pretty well on course to solve a lot of problems here in the future. So as I mentioned, in the same way that we would monitor groundwater at a contaminated site, it's important for all of us to look back and see how far we've come to trend our data. And so over the past 30 years, you've worked with us and you've helped us, and you've also seen us breaking apart the black boxes of many different industries: contaminated groundwater, chlorinated solvents, petroleum hydrocarbon, corrosion, and now we're starting to dig in to some of the black boxes of the future. And so I want to thank all of you for your time. And no matter what we're looking at, whether it's the subsurface or it's our reservoirs or our pipelines, whether it's our farmers' fields or it's our groundwater, we're going to be working with you for the next 30 years, breaking apart additional black boxes and staying on course to continue to move forward in our industries. So with that, we want to thank you all so much for being a part of this webinar, and we're going to answer any questions that you may have.
All right, thank you so much, Sam and Kate. as you can tell, we are super excited about where we've come from, what we're currently doing, and where we're going. And I hope that you found something innovative and interesting in this presentation. We do have quite a few questions, so we'll try to get through a few of these. If you have others, again, just enter them in that question box on your GoToWebinar panel. One of the questions that we have that's really simple that I'll start off with here is it possible to have a copy PDF of the presentation? And we're happy to provide that. Just send an email to info@microbe.com and we'll be glad to send that over to you. The first question, Sam, I'm going to throw this over to you. Can you talk about how we sample for the new meta array analysis?
Absolutely. So if anyone has performed sampling for any of our qPCR analysis or any of our gene-based analyses using our bioflow filters, it's going to be the same. You're going to use a low flow pump and push water from wherever you're sampling through a bioflow filter, and then that's going to be shipped to us on ice. So we're happy to always send any type of sampling protocol, and we provide those sampling supplies free of charge.
Awesome. All right, next one is for you, Kate. We know that benzene's often the driver for LNAPL cleanup. So can you talk a little bit about the aerobic genes that we've identified and maybe some of the research that's being done and, about kind of what you know about anaerobic stimulation of benzene degraders?
Sure. For anaerobic benzene degradation, we have one main qPCR assay that we're looking at for that is the anaerobic benzene carboxylase gene. And I believe I don't have the exact detection frequency, but I do know that it is detected less frequently than the BSSA gene that's for the anaerobic text degradation that Dora was talking about. So it is not as common at sites. So we can look for qPCR, with that qPCR assay. We can also use stable isotope probing to confirm benzene degradation in your current environment. And so that method, it does not distinguish between aerobic or anaerobic degradation as long as the benzene is being degraded and incorporated into the biomass or carbon dioxide, then we can detect degradation that way too. Stimulating anaerobic benzene degradation, we have had some clients who add sulfate or nitrate to help with that process.
All right, thank you, Kate. Sam, can you talk a little bit about the mitigation approaches for MIC that MI monitors what's happening and kind of what our involvement and role in that is?
Yeah, so we have a number of different methods that we can use to look for key targets related to microbiologically influenced corrosion. In terms of mitigation approaches, there are a number out there. There's consistent approaches, things like pigging or using biocides. And the way that we would work into that format is to do pre and post. So again, we're trending the data. We're looking at whether the biocide is actively working, whether the pigging is removing biofilms and we're seeing fewer. We can be used within regular analysis, things like coupon analysis that can be incorporated within the pipelines. There's a number of different ways that our tools, our current tools, can be used to help in terms of mitigation and tracking what the vendors provide. And we normally talk about this and we haven't yet, but I want to remind everyone that we are strictly an analytical lab. We do not provide any kind of, we don't sell any kind of products. We're not a vendor, but we'll work with just about any vendor that you have. So that allows us to be unbiased in our analysis.
Yeah, and to add to that, Sam mentioned trending the data. That's really important in these systems so that we can understand when a biocide has been applied in that system, what the flux in the microbial concentrations are, because you can sometimes see like a rapid decrease in certain groups and an increase in other groups that can be just as corrosive. So you want to make sure that you're monitoring, like using the QuantArray MIC to be able to monitor those to see how the overall community is changing, and then do that over time, because as the systems adapt to these treatments that are being done, you need to make sure that we understand that adaptation that's occurring. So just a side thought on that. Okay, next question, Kate. When you have a site where we don't have, historical microbial results to rely on, where does this person start? And they are listing that the contaminant of concern is 1,4-dioxane. So can you talk about how you would advise someone for an initial site assessment?
Sure. A great place to start is with qPCR analysis to see what the genetic potential is under your current conditions, what microorganisms are present. And we do have several different assays that are related to 1,4-dioxane biodegradation. We can track some genes from pseudonocardia CB1190, and that's a strain that Dr. Shaylee Mahendra's group has studied in depth, and that we can track their genes because they can do the direct metabolism of 1,4-dioxane. We also have some aerobic co-metabolic genes that could be helpful for 1,4-dioxane. So contacting us to find whatever your contaminant is you're starting with, we can recommend which qPCR assays would be helpful to start to get that baseline to see what you're working with.
Awesome. Thank you, Kate. All right, Sam, when you're talking about that meta-array data that we saw in that site, what do you think was going on between the samples that had the low reductive dechlorination potential and we're only showing moderate Dihalococoides counts? Can you explain that?
Yeah, that's a great question. So for that specific site, we're waiting on additional data to start to dig in and see why. And I think that's why we are especially excited about the meta array and being able to use it as a screening tool to see what locations are showing low potential. That way we can dig in a little deeper with additional tools, or the site manager can dig in a little deeper with additional tools. Some of those things might be the overall redox conditions of that area. They might be nutrient availability. It could even be things like pH. And I also think it's important, again, we keep coming back to this, but it's going to be important to trend that data. So if we see that perhaps Dihalococoides only reached that region recently, then the next time that it's sampled, we might see it starting to move more towards the high reductive dechlorination potential.
Yeah, and this data really excited when Sam and I were kind of reviewing it with the client, because I've seen this for so many years where it's like you get to your next round of Dihalococoides sampling and you got several that are in that 10 to the 3 group where it's good, but you're not exceeding that 10 to the 4 or 10 to the 5 threshold where you want to be. And we would see in the next round of sampling, some of them made it there and others didn't. And we couldn't figure out what was going on. Well, metabolites help us to understand that. And as this tool grows and we have more knowledge of what these metabolites mean, I think it's going to be even more predictive. So imagine if you didn't wait another quarter to find out that some of those weren't going to make it where you wanted to go. You could start being proactive, and again, that 1 degree of change a lot earlier in site assessments. I think it's really exciting data, and we're definitely interested in sites to utilize this tool. So reach out to Kate if you have a site that you think would be a good fit for it, and let's talk through kind of the data and what we can do with it currently and where we want to go. And we are looking for more research in that. I think you're going to see over the next year or two this expanding, as Sam was mentioning, into petroleum hydrocarbons, potentially into PFAS, as well as to other markets. So it's a really exciting technology to have. One final question before we end today, and I think it's a good one. This asks if we collaborate with people that start up companies to do different monitoring, and we certainly do that. Sometimes people are developing tools, like in particular, this company is doing an on-site electrochemical technology to look at rates of MIC and abiotic corrosion. And so if you have things like that that you're interested in, we're definitely interested in helping. We certainly are partnering with companies now in the carbon sequestration space and in applied genomics, where they're doing some really interesting things for genomic changes for site assessment. And we're already working with those companies to develop qPCR assays specific for the products and processes that they're interested in. So we're always open to that. If it is something that you're interested in, you can can reach out to Sam Rosalina, and he would be happy to give you some more information. If you don't have his email address, just email info@microbe.com and they'll get that over to Sam so he can reach out and talk to you. Again, thank you, Sam and Kate, for working with me to put this presentation together. It was so much fun to put this and to think about where we are and where we've been and where we're going with you guys. And so I really appreciate the collaboration that we have. And thank you all for being a support for Microbial Insights over the past 30 years and for being here with us for this presentation today. We are very grateful to our customers and our colleagues that have supported us all these years. And we are looking forward to another 30 years plus to work with you guys. So I hope you have a wonderful day and we appreciate you joining.